Hello, I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get accepted into medical school and other professional programs. And welcome back to another episode of MCAT Bites. Today we're going to delve deeply into the structure and function of proteins and their constituent amino acids. This topic is fundamental for understanding how proteins mediate the behavior of biomolecules, which is a key concept for the MCAT Biochem and ChemPhys sections. By the end of this video, you'll have a solid grasp of amino acid structure, protein folding, and the diverse functions of proteins in the body. Let's get started. And quick aside, if you are keeping track, this would be going over all of MCAT Foundational Concept 1A. All right, with that out of the way, what's an amino acid? It is the building block of a protein. Each amino acid consists of a central carbon atom, shown in the center, good place for it, known as the alpha carbon, not a beta carbon or a gigacarbon, but an alpha carbon. It is bonded to an amino group, an NH2, a carboxyl group, a COOH, and a hydrogen atom, and a unique side chain, the R group. This is what makes the amino acid special. The 20 standard amino acids have different properties based on the R group, which can be polar, nonpolar, acidic, basic, chiral, achiral. These properties influence how amino acids interact and form proteins. There are a lot of different types of amino acids, and this is definitely something that you won't memorize from this video, but you should memorize after this video. So grab some screenshots and make some Anki cards, because this will take a bit of work, but if you don't get this memorized, you will be missing a ton of easy questions on the MCAT. So you need to memorize this table. But let's go over it briefly here. Our nonpolar amino acids have hydrophobic side chains. Examples include glycine, alanine, valine, leucine, isoleucine, methionine, phenylalanine, tryptophan, and proline. These amino acids tend to be located on the interior of proteins away from the aqueous environment, whereas polar amino acids, that's the opposite. They participate in hydrogen bonding and are found on the exterior of proteins interacting with the aqueous environment. These proteins include serine, threonine, cysteine, tyrosine, asparagine, and glutamine. And we can look for some patterns here. For the polar amino acids, notice there are a bunch of hydroxyl groups. We've got OHs. And if there isn't an OH, we've got these carbons bound to a double bond oxygen with an NH2 attached. Next, we've got our acidic and basic amino acids, which I should note are also polar, but they have side chains that can donate a proton for the acidic ones. Examples are aspartic acid and glutamic acid. It's nice that it's in the name. They carry a negative charge at physiological pH and often play a role in enzymatic active sites. Whereas basic amino acids have side chains that can accept a proton, i.e. they're negative. Examples are lysine, arginine, and histidine. They carry a positive charge at physiological pH and are often involved in interactions with negatively charged molecules like DNA. Amino acids can be linked together by peptide bonds to form polypeptides. This just means a bunch of peptides. A peptide bond is formed between the carbonyl group of one amino acid and the amino group of another amino acid through a dehydration reaction. This reaction releases a molecule of water. The sequence of amino acids in a polypeptide is known as its primary structure, and this sequence determines the protein's final three-dimensional shape and function. But we need to know all levels of protein structure. Primary, we just talked about, it's literally just the sequence. Secondary is the local folding pattern, such as alpha helices and beta sheets, stabilized by hydrogen bonds between the backbone atoms of the polypeptide chain. Alpha helices are right-handed coils, while beta sheets are formed by linking two or more peptide chains side by side. The tertiary structure is the overall three-dimensional shape of a single polypeptide chain. Key on single polypeptide chain, stabilized by interactions between side chains, including hydrogen bonds, ionic bonds, hydrophobic interactions, disulfide bonds. The structure is crucial for the functionality. Now, quaternary structure, not all proteins will have, but they are an assembly of multiple polypeptide chains. Keyword multiple. Tertiary, this is just one continuous string. Quaternary, we've got a bunch of different continuous strings. That means they have subunits, which is a functional protein complex. Examples include hemoglobin, which consists of 4 subunits, and DNA polymerase, which has multiple subunits working together. On to protein folding and stability. Protein folding is a critical process that determines a protein's function. Proper folding is guided by chaperone proteins, which help the nascent polypeptide chain to fold correctly and prevent misfolding. Protein stability is maintained by various interactions, including hydrogen bonds, ionic bonds, hydrophobic interactions, and disulfide bridges. Our tertiary structure we talked about before. Misfolded proteins can lead to diseases such as Alzheimer's, Parkinson's, and cystic fibrosis, where the loss of proper protein function has severe consequences. There are a lot of different functions of proteins, from enzymes, which catalyze biochemical reactions, to structural proteins, which provide shape and structure to tissues, to transport proteins, which carry molecules across cell membranes, signaling proteins, which signal between and within cells, and motor proteins, which lets us move around. A lot of different proteins. Understanding the structure and function of proteins and their constituent amino acids is fundamental for mastering biochemistry and cell biology. Proteins are versatile molecules that perform a wide range of functions, from catalyzing reactions to providing structural support. Keep studying these concepts and you'll be well prepared for the MCAT and your future medical career. Thanks for watching, and see you next time on MCAT Bites.